Welcome. My name is Helen May-Simera, and I'm a professor at the Johannes Gutenberg University in Mainz, Germany. I'm really grateful that I've been given this opportunity to introduce myself and the work that I do, which is all centered around a better understanding of what causes Bardet-Biedl syndrome. So who am I? Like I said, I work at the Johannes Gutenberg University, which is the largest campus university in Germany, and it's in Mainz. Mainz is a beautiful town along the Rhine, which is one of the largest rivers in Germany, pretty much in the center of the country. But how did I end up here? So I've been involved with the BBS community since 2003, when I started working on my PhD with Professor Phil Beals in his lab at the Institute of Child Health. So this is a pretty ugly building in the center of London, and it's both part of University College London, UCL, and Great Ormond Street Hospital for Children, GOSH. So back in 2003, when I was looking for a laboratory to do my PhD in, I had never even heard of Bardet-Biedl syndrome or BBS. But I have to say that Phil seemed like a really nice guy. He seemed very approachable, and he was also really excited about the projects available in his lab, especially because they had just published the first paper about the underlying causes of BBS and connecting it to cilia function, and in particular to the primary cilium. So I'm sure you're all aware, primary cilia are these tiny little hair-like structures found on the surface of almost all cells, kind of like a candle on a birthday cake. And so here in the middle picture top, there's a cross-section cartoon of a primary cilium, and underneath that, this grey, black and white image is a real picture of a primary cilium that I took myself on an electron microscope. And the kind of tiny little wiggly thing sticking up there, that is the primary cilium. But back in 2003, the function of this structure was unknown. And how it worked was all still very much a mystery, which made it a really exciting topic to work on. The other reason that I was really keen to work on BBS was the chance to get to know the patients and the patient community. So Phil had promised me that I would be able to come along to the yearly conferences held by BBS UK in Northampton each year. So Northampton is also pretty much in the centre of the UK, and people from all over the country meet once a year. And I've really made it my mission to attend each one of these conferences since I first joined. So my first conference was in 2004, and I think since then I've only really missed 2 that I can remember. And it's this close connection to the patient community that made me even more determined to understand what causes BBS and what could be done to help the individuals affected by the syndrome. So in the lab back in 2003, we were starting to figure out that primary cilia are used by cells to communicate with each other. More specifically, they're able to receive signals kind of like a satellite antenna. And this communication between cells is important for all sorts of functions in the human body. However, in some organs and tissues, these processes are even more important than others. For example, in the eye or in the kidney, there's a special subset of ciliated cells that really need to communicate and talk to each other. And that is also why in individuals with BBS, they sometimes have problems with these tissues, because these tissues have specialized ciliated cells. So during my PhD, I really became fascinated to try and understand how cilia help these different organs develop and function. And without really understanding what is going on a molecular level, we cannot begin to properly design treatments and treatment options and different strategies. So really getting to the bottom of how the different BBS genes and proteins are involved, not only in the cilium, but also in other places. It's really vital to understand so that we can start developing, mistake. I want to go back, edit. So really getting to the bottom of how these different BBS genes and proteins are involved in these processes is a vital part of medical research. And this is not just on a cellular level, but also on a molecular level. So although we've made some progress during my PhD, the more we learned about how these BBS proteins work, the more questions arose. And so after my PhD, there was no way I could stop. I was given an amazing opportunity to move to the US and to work on these questions at the National Institute of Health, just outside of Washington, DC. And I jumped at this chance. So this is one of the largest medical research institutes in the world. And here I really was able to start working more on why cilia don't work properly in people affected with BBS and what specifically is causing these problems. And after several years in the US, I was extremely fortunate to be able to get a grant to start my own lab dedicated to answering these questions. And that's why I moved to Mainz, Germany. And at the Faculty of Biology at the university here, I was able to set up my own lab. So my lab, we're working on 3 main research themes, all trying to solve the puzzle that is BBS. One of the first main questions is, how do the BBS molecules help ciliated cells communicate? One of the second questions is, why does defective communication between these ciliated cells cause problems, particularly in the eye and the kidney? And one of the third questions is whether the BBS proteins actually have alternative functions and maybe do something else, not just in the primary cilium. And in the next few slides, I want to very briefly try and explain a little about what we found out and how that's relevant to the BBS community. So the first story I want to share with you is about how the BBS molecules help cells communicate. We used to think that the primary cilium is only there to receive signals and information from other cells. But now we know that cilia are also able to send out parcels to communicate. Now these parcels are called extracellular vesicles, or EVs for short. And these are actually membrane-bound packages that essentially contain signaling molecules. It can be a variety of different molecules, for example, proteins or DNA or different lipids. And the cells can actually communicate by this kind of EV-mediated postal service. So to be able to study this form of communication, we need to come up with a way of collecting these EVs, or so-called parcels. And there's actually several ways we can do this. So first of all, one way to collect these is directly from bodily fluids. So EVs are found in all bodily fluids. So blood, saliva, breast milk, and particularly in urine. This is really helpful and useful for us because the ciliated cells in our kidneys actually release these EVs and they then end up in our urine. So after collecting urine, there are several ways that we can clean up and concentrate these particles. One way we can do this is centrifugation. So with that, you take the urine, spin it very, 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 very fast. And then if you're lucky, you can actually get them pelleting out at the bottom. The other way is to use very, very fine sieve. So this is way, way finer than the sieve that you'd use in your kitchen. And you actually make this sieve out of small agarose beads. And that way you can also kind of get them, filter them out. The other way we can also get these EVs is that you can collect them directly from specific cells grown in a dish. So what we can do, we can actually grow specific types of cells. So we could use skin cells or kidney cells or other types of cells, or cells from the eye, or maybe specific cell types from the kidney. And we can grow these cells in a dish. And when cells are grown in the dish, they're covered with a liquid called media. And this media, this liquid media, contains all the growth factors and the nutrients that these cells need to survive. And so these cells will also release their EVs into this media. And so over a collection period of normally 24 to 48 hours, we can then harvest this media into which these EVs have been released, and then isolate them again in the same way as we did with the urine, either by centrifugation or by filtering. And the great thing about this is that we can actually generate specific cell type specific EVs and have a look at them and compare them. And when we actually look at them in more detail, one of the ways to do this is to first of all take a picture and have a look. And for this, we need a special type of microscope, electron microscope, because these are really tiny. So some of these vesicles can be only 100 to 150 nanometers in diameter. And if you look at them underneath an electron microscope, they kind of look a little bit like a deflated soccer ball. But to be honest, looking at them doesn't really tell us much. What we really need to know is what's inside these EV particles, these EV parcels, because what we need to know is what's inside them so that we can figure out what it is that the cells are actually trying to tell each other. And that is what we're trying to do right now in my lab. So we are only at the very beginning of our investigations, but we've already found out a few key points that I want to highlight here. So first of all, we know that when the psyllium doesn't work very well, this parcel service is disrupted. So this means that a dysfunctional cilia can affect the release of EVs. So it seems that we're getting more EVs when the psyllium doesn't function very well, and also we get different types of EVs, so different types of parcels and packages that are being released. And it's not just that, it's also the content of the parcels appears to be different when the function of the psyllium is disrupted, as in the case of the BBS patients. So not just the number and the type, but also what's inside them. So the other thing that's really exciting is that these parcels, they don't just look pretty, but they're actually bioactive. And that means that they can actually do something. So if you take out these EVs and take them from one cell, and actually if we give them to another cell, that can initiate a response, as can be seen here depicted by kind of glowing stars, because we can actually make that cell do something. And if we use a special reporter cell, this reporter cell can then light up. And this means that these are really actually important and will actually be taken up by other cells and are not just a waste product. And again, this is something that's really quite new, and we're just trying to figure out what this actually means for the communication between cells and what it actually means for the different organs and tissues that we are working at in and interested in. And like I said, it's important because it really helps us to expand our knowledge of how these cells are beginning to communicate. And it could also help us to explain why different cells in the body in certain tissues, such as in the eye and in the kidney, aren't working as effectively as they could be in a BBS patient. And that's why gaining new insights into these molecular mechanisms can really help guide the development of novel therapeutic approaches. Now, very briefly, I just want to address a second topic that we focus on in our lab. And that is the fact and whether BBS proteins might have alternative functions. So for a very long time, we thought that the BBS genes and proteins were only required in the psyllium. Now we know that that's not the case, and that they can also be found at other places in the cell. And one of these places is the nucleus. So the nucleus is depicted here as a round kind of blue watermelon. And the nucleus is the region in the cell which is where all the DNA is stored. And it's what controls and regulates all the activities of a cell. It's a little bit like the command unit on a spaceship. So this is really very new. And what we're trying to figure out is exactly which BBS proteins can enter the nucleus and under what conditions. Because that really is not clear and no one's really been looking at that yet. So what they're doing is really not clear, but it's obviously that if they're going there, I'm sure they're doing something. And so this is what we're trying to figure out in the next few years, especially since these alternative functions away from the cilium and somewhere else in the cell, they are very, very likely to actually affect the symptoms that an individual with BBS might be having. So obviously we want to try and figure out what they're doing there and why. So as you might have noticed, the more we learn, the more we find that there's left to discover. And all of this research cannot be done in isolation. So the BBS research community, and especially also the wider cilia community, has already expanded exponentially in the last few decades. This growth can be measured by the number of publications involving BBS. And as you can see from this graph here, which shows the number of publications, the total being 1,409, from 1946 to 2022. And this was actually taken on the 21st of June of this year, 2022. And as you can see, in the past decade or so, there's really been an explosion of publications, scientific publications involving BBS. And there have been some fantastic advances in diagnostics and also in development of possible treatments such as gene therapy and appetite regulators. But we still don't have all the puzzle pieces. There are still lots of open questions remaining. And only when we really, really understand all the different aspects as to why the symptoms occur in affected individuals, can we really begin to find lasting solutions. And so as a community, we really need to keep growing. And I can see this happening in a few ways. So first of all, a continued foraging of connections between the different research groups around the world. And this is absolutely vital, not just sharing knowledge, but sharing resources. And I have to say that I think on this front, the corona pandemic has actually sparked more online exchange and debate. I think we all kind of realized how much we can actually do online. And this has really brought about kind of a big change with more groups sharing lab meetings online across different boundaries and different countries that maybe before one would not really have thought of. And so in that sense, it's been a kind of a silver lining of the pandemic. There are also increasingly more meetings and scientific conventions going on involving cilia scientists, which is obviously always a really good thing. Secondly, I think it's really, really important to foster the next generation of research scientists. There's only so much we can do, and we need to keep expanding, and we can only do that by getting young people excited about this research. So I'm very fortunate because being in an academic setting, it allows me to recruit lots of bright and talented individuals and get them hooked on this really, really fascinating field of research. And I'm really, really proud because I've got several members in my lab that they themselves have gone out and gotten their own funding because they're really keen and dedicated and want to keep working on the projects that they've been starting to work on during their studies. And the other aspect is for this, I think the connection and the affiliation with the patient groups is incredibly important because it's incredibly rewarding for the students. It really, really makes a lasting impression when the students can interact with an individual that has BBS, and it makes them more motivated in the lab. And so I really try to get my patients and my students involved in hosting workshops at different patient conferences, and really getting out there and trying to communicate the work that they do. And I think this actually goes both ways, this connection, because I also think it's absolutely vital for the patient community to learn more about what's happening in the scientific community. And this can be done in different ways, for example, with outreach events, online information sessions, or by hosting lab visits or workshops to the lab. I think this dialogue between researchers and patients is vital, as it can not only motivate the students, motivate the researchers, but it also can help direct the research needs to show where it needs to be going in order to find ways to improve the lives of those living with the syndrome. Now, with that, I'd really like to close. I want to thank all of you for listening. I really want to thank BBS International for giving me this platform to showcase our work. I want to thank the BBS community for giving me and my team endless encouragement and motivation to keep going. And I really do hope that I'll get to meet some of you at a conference in person one day. So thank you again, and goodbye.